Dutch pronunciation is van het Veer, but actually Veer. So I'm happy to tell you something about what we are doing at UCSF, and it's a large consortium working on molecular and clinical data actually in the real-time breast cancer world, and how we are moving all of the molecular data and computational work we're doing into the patient care arena. So just like a very brief introduction, the biological types of breast cancer, of course, help us to tailor treatment. And we are particularly interested for those breast cancer patients that have a high risk of recurrence. And maybe related to the first speaker, I think the more you can zoom in to very specific types of a cancer type, the better I think all of your signatures will perform, because not everything is the same. So breast cancer originates in the epithelium of the terminal duct of the lobular unit, as you can see here. And is, of course, an interplay between genetic makeup of the host, physical tension, microenvironment, and other exposures. So each of those cancers come with a different risk factor profile, and therefore also have a different biological makeup of the tumor itself. For many, many years, diagnostics in the clinic have been done by just single gene immunohistochemistry, but of course it doesn't appreciate the full picture of a tumor. And now with all our genomics efforts, we're much better to dive into the deep. It all originated from work at Stanford from Chuck Peru and Teresa Surley by looking into the first gene expression microarrays to understand that if you take a section of a tumor, and in this case tumor plus microenvironment, because I think both are very relevant to foretell the nature of that tumor, that you can in the gene expression distinguish all of the different gene expression modules, and that led them to propose, based on the biology, 5 different subtypes, which are often used and got a little bit more in other research up to like 10 subtypes, but this is sort of the basic that is still being used. I worked myself on a prognostic signature, which he developed to really look into the signature that could foretell outcome of disease of a breast cancer patient, and came up with 70 genes that can foretell high risk or low risk of developing a metastasis, which is currently used in the clinic and FDA cleared. So here what I want to tell you about those women that are high risk of recurrence, and to see how can we actually optimize their treatment, because if they have a high risk, we should target their treatment. So many of you are probably aware of this particular diagram, first postulated by Weinberg and Hanahan, which is actually often shown that the biological processes in cancer are deregulating and changing replicative immortality, tumor-promoting inflammation. And so there are over 1,000 targeted drugs in development, and maybe less than 50 being approved for certain indications to actually help target cancer. And it's a way of how do we efficiently combine these therapeutics to efficiently kill the tumor cells. And there's, of course, genomics very helpful. So in case of high-risk breast cancer, what is studied at the moment is targeting the tumor immune environment. For many, many years, people have been looking at immune environment, but only most recently understanding what are the relevant targets to hit, which the ones down here, the PD-1, PD-L1 receptor and ligand is the most effective at the moment. But there are others that actually target particular types of cells in the microenvironment of a tumor tissue. MYC is one of the genes, stem cells, certain PI3 kinase pathways, and the HER2 families, and there are many more. So you need to come up with a system to efficiently, in patients, actually test all these drugs, because it had been calculated if you take all if you take 1,000 targeted drugs under development, and if you envision that it's a combination of 2 that will be most optimal, we need to have, and you have to run a trial of 1,000 patients for each, then you would need 5 billion patients to actually test all those drugs in clinical trials. So it's not, and those 1,000 patients are needed if you take a 5-year or 10-year outcome. So that would take much, much too long. So we need to move to inpatient response monitoring, and also in a much shorter time frame, so that within a few months you actually know whether there is a response or not. That also comes with a risk, because you're not really immediately evaluating long-term outcome. But I think if you want to get to this combination of drugs, we need to come up with clever endpoints, which also includes actually imaging.
Question?
Yes.
Immune environment. Do you need to know and sequence immune repertoire, or you can have it?
People do it in different ways, so it's actually still unclear. So currently, the drugs that are targeting this PD-1, PD-L1 receptor ligand are actually given to all patients in melanoma, or all patients in lung cancer, or all in breast cancer, and people see responses. Not in everybody, and so they're developing the test to look into whether the T-cell repertoire is actually a biomarker that can help facilitate that indication. But it's sort of moving on in parallel. It's all in clinical trials, and I think the I-SPY 2 trial that I want to discuss, we actually set up 5 years ago, so that while a drug is being tested, we qualify biomarkers at the same time under a high stringency. Because besides these 5 billion patients that need to be in clinical trials, if you would have a long-term outcome perspective, you also need to set up a system where your stringency in testing your genomics is at high quality level. So then you can actually also at the same time test the T-cell repertoire or other molecules in that context. So this is a trial that originated from UCSF. Laura Esserman, who is the surgeon, and Don Barry, who's at MD Anderson and who runs many Bayesian probability clinical trial schemes, and one is an adaptive clinical trial. So I'll show you how we are trying to, during the trial already, try to enrich the patient population for a particular arm that seems to respond. So if you may know about HER2-positive breast cancer and the drug Herceptin, which is approved for many, many years. So if you would take all breast cancer patients and 15% is HER2-positive, if you would start to measure HER2 and have Herceptin, then after some 20 patients, our system would indicate that the HER2-positive ones are the ones that are responding, and then the next patient who is HER2-positive would go into that arm of the trial, and in that way we are enriching for the patients who respond. So we select the high-risk patients by looking into the biology using this 70-gene prognostic signature, and if that is high, then those patients are randomized, first of all, on biomarker signatures that use the estrogen, progesterone, HER2, and the MammaPrint score in another high-risk 1 and high-risk 2 part. So here is a list of drugs, new agents that we're testing in this newly diagnosed patient population, and you can see that different targets are listed in the middle column, and the one in purple are the one that we actually either successfully graduated or one was dropped for futility. The ones that are in white are currently in the trial, and 7 agents are in the pipeline. Actually, if one comes out of the trial, a new one can come in. So this is all one trial process, which is now running for 5 years, and every new drug only needs an amendment to get into the trial. So this is how it works. There is one standard arm, standard of care, and there are up to 5 experimental arms where the investigational agent is given together with standard of care. Patients are being diagnosed, so this is their first breast cancer, evaluated by imaging, and a biopsy is taken, and then ongoing there are imaging MRIs as well as biopsies or blood draw, and the endpoint is surgery, and this time frame is 6 months. So that is a much shorter time frame than, of course, 5 years or 10 years. So we recognize patients can be HER2 positive or negative, and in each of those there are multiple arms. We take the MRIs and biopsies, and every patient informs actually the system. And once we reach a statistical significance, and I'll show you how we do that, then the the drugs can go out of the trial and a new one will come in. So in a simplistic way, in the start of the trial, the learning part, patients are randomly assigned to an arm. We determine based on their pre-treatment biopsy, their biology based on just a few simple markers so far. And then we recognize 8 different types here indicated by purple, blue, and yellowish. And we then, at time of surgery, the pathologist will determine whether there's tumor cells left or not. And if there is no tumor cells left, indicated by an X, this is called pathological complete remission. And actually, remarkably, after 3 cycles of therapy, so that's really early on in the trial, so at this earliest time point, already 30% of the patients have no sign of tumor left. So some of these drugs are very efficient, and even after 3 cycles, so this is a few weeks, you can already identify who are the ones who will likely end up without tumor at surgery. So we're actually evaluating that so that in future time we can start to change therapeutics even early on.
Question?
Mm-hmm.
Surgery is done to all? There's no... What about cases of remission? Do you eventually have
Of recurrence, you mean? Okay. So these are newly diagnosed patients, so the tumor remains in the breast and we're following it for 6 months. And so we're looking at shrinkage of the tumor or not. And then there's surgery. So the tumor is...
Are all undergoing surgery?
Yes, all are... There are actually a few patients when there's really, after 3 cycles, no tumor left and there's no tumor on their pre-surgery MRI. There are a few patients who have opted to do no surgery at all. And some of our previous trial actually are still disease-free, but it's like by no way a standard of care. So this is, you start to assign randomly, and at one point you start to recognize that biology type 2 responds to drug 1, so you start to enrich for that particular biological subtype in that arm, assess the PCR, and when there is a predicted likelihood of a success in a subsequent 300 patient phase 3 trial, then the drug can graduate out of the trial. And it's anticipated that you only need 100 to 120 patients to find a successful drug biomarker combination or a failure. And actually our first graduation was after even only 72 patients. So you have less patients, a shorter time frame, and so you're set up to earlier graduate drugs to a next phase. And the FDA actually recognized that for high-risk breast cancer, this is a very relevant endpoint. So they now allow, partly based on what we have built with I-SPY2, that is a valid approval status. So we begin the trial with equal randomization probability, calculate success for each of the biomarker signatures. Then for every patient that goes to that, we run again a graduation or futility. And then if it's above a certain threshold of this success in a next trial, then there is a stop of accrual in that arm. Or if there are too many patients, it's stopped because it will never be better. And then a new drug will enter the trial. So I'll show you, just to dive a little bit deeper into the biomarkers and genomics, one of the first arms, which was a PARP inhibitor, so that's a DNA repair inhibitor. 72 patients were randomized to get these drugs, and 62 patients were concurrently to the standard regimen, so those are the controls. 44 are HER2 negative, and those are actually the ones that are being evaluated against the drug. So this is the proportion of patients in each of the HER2 negative groups that were treated, that were HER2 negative, hormone receptor positive or negative, and mamaprint high 1 or high 2, which is ultra high. Yes.
Do you have the BRCA status of these patients?
We do, and I will come to that in a minute. That's actually a minority. So this drug was given to all HER2 negative patients, and you can see in all patients it does a little bit better than the control. If you look actually at the triple negative group, it does twice as well as the control. whereas the other group, the one that is hormone receptor positive, it doesn't really do better. So this is the signature for which it was enriched, and it shows high probability of success in the next trial. So we identified the biomarker signature based on just only hormone receptor and HER2, those had to be negative. But if you look at the data, then even in that group, it's only 52% who respond, so that's still not everybody. So there's room for improvement. So we started to look in what is actually the biology of those patients that are responding, whereas in parallel, this particular combination is now being tested in phase 3 for just the triple negative patient group. So that means, and that trial concluded accrual, so that means that from the start we have now a 4-year time frame and I think they will need another year follow-up, so 5 years until we would know whether this drug combination can be accepted by the FDA. So already pointed out there's 52 response, so how about other biological pathways? And we're set up to actually qualify biomarkers, and there comes all these DNA repair and BRCA status. So those are hypothesis testing, which I will show you some of the results. Whereas we're also going into the exploratory phase where we're just going to analyze all of our next generation sequencing phosphoprotein data to even do better. So we have of all these patients, so 1,700 have been screened, almost 800 are up to surgery, 1,000 went into the trial. So we have more than 30,000 biospecimen files over these different time points, and are either tumor biopsies or plasma serum or Buffy-coat to test and to look into all the biomarkers. So that means that every patient has 3 times that biopsy cores are taken from the tumor, which are like tiny, tiny cores. So they're 1,000 micron, and we are able to actually section from all of these sections by microtome, frozen, and we can do for most patients all of these assays out of one of those tiny cores. Sometimes we need a second one, and we qualify those that they need to have more than 30% tumor cells. So we have a wealth of data starting to accumulate to further test. So how about these 52% of patients who responded to this PARP inhibitor? The PARP inhibition works on the DNA repair. So if you further inhibit, then you assume that if you are doing that efficiently and the tumor cell is indeed dependent on this DNA repair deficiency, then you will be able to have the cancer cell die and the patient will get a pathological complete remission. So we tested either in the germline, which is number 1, the BRCA1 and 2 status, which is of course the hallmark of patients having a DNA repair deficiency intrinsically, and number 2 to 5 are actually assays we did on the tumor itself to see if we could find signals for DNA repair deficiency, either by a gene expression profile we developed for PARP inhibition based on cell line data in the past, or a BRCA signature, for instance, which can identify DNA repair deficient cells. And we actually also used the ultra high risk signature, so highly proliferating cells. So it's a very complicated scheme, and I can go into details later, but we have a 3-step process where first we look into the relative performance of such a signature in the treated patients versus the control. So it needs to do better in the treated patients than in the control, because then it's associated. If this is passed, then we evaluate it in the context of in this case, the triple negative graduating signature. If that passes, then we go to step 3 and we do an actual Bayesian analysis as in the randomization engine. Because it's an enrichment trial, you need to do a Bayesian analysis to actually be able to compare apples to apples in this case. So the association with response, there is a threshold of a p-value of lower than 0.05, and each of these that have a yes passed it. And so you can see that the BRCA mutation status passed within the VC arm, but not as a interaction, because there are actually too few BRCA mutated patients in the whole trial. So I think it's still a valid biomarker, but our group was too small.
So that's just germline status. You didn't sequence the BRCA in the tumor just for somatic?
This is germline mutation testing, which was performed by Myriad Genetics.
But do you have somatic status in your, because
We are actually now also developing mutations in the tumor tissue itself, but so far it was only germline. So we looked at this PARP inhibition and BRCA signature, and you can see if you adjust for subtype, in this case triple negative, this PARP inhibition and the BRCA status is significant. And in the Bayesian, I realize I'm showing you many different types of analyses. So Bayesian analysis gives you a probability of pathological complete response. So I showed you before the 52 versus the 26%. Since we're adding a few more markers in this particular analysis of all triple negative, it is 56 versus 27. If you then take the ones that are PARP inhibition signature high, you can increase, which is 65% of this group, you can increase this probability to 69%. If you take the very high proliferating tumors, you increase it to 64%. And if you combine it, you increase it to 79%. So what we effectively have done is that, again, back to this unselected group, if you take the 40% of this group that have both markers, we can predict 80% almost response rate, which is really, really high, whereas the other 60%, there's hardly any response. So we're actually pretty excited about this result by homing in on who has what biology and having a real stringent process. I don't know whether any of you have ever worked with Don Barry, but to get something like this that takes for him to approve what to show takes several weeks. But anyway, I think we have a very stringent process, and I'm excited that really the biology can help you to zoom in, and that's further detailed here. The estimated PCR rates here in numbers again goes from 56 to 79, and the probability of success in a phase 3 trial would still be 99%. So this particular one in all triple negative is currently conducted by Abbott, who is the owner of this PARP inhibition. So we hopefully can test our biomarker combination to see if we can identify there again the ones that truly respond. So we screen in I-SPY 2 patients with high risk, more proliferative tumors where this pathological complete response is predictive for therapy response, and actually we know is also predictive for long-term outcome in general. This accelerates the process of identifying drugs that are effective in specific breast cancer subtypes, and it dramatically reduces cost, time and number of patients to get these drugs to the market, because the FDA has given this an accelerated approval status. So we are working with 10 different pharma companies who all want to test their drugs in this particular setting. They all run their own trials as well, but this is one of them. One step further into the genomics and computation, which we're actually starting up right now, is in these exploratory biomarkers, where this is the integrated data systems that we have. So the whole I-SPY clinical trial runs on a Salesforce system with all of these different data types. There's a lot of molecular data that all sits either in a specific system or also in hospital systems like sequencing. So we're currently now deposited the first data on the supercomputer at San Diego, and we're working with the BD2K and Global Alliance Group to use APIs to actually efficiently interrogate this data, but actually building this integration layer here. I'm not the expert, but I talked to many people, and I think nobody really has built this efficient integration layer. So we're constantly talking to different people, and I think the challenge is to find a combination of experts from different companies, from different academia, to actually really build this part efficiently. So if you have ideas, then I'll be happy to talk to you. The last part is maybe also this particular here on the bottom that I didn't allude to, but as we are testing biomarkers in a stringent way, actually that facilitates that we can do that under a particular FDA approved system, which is called the IDE, an Investigational Device Exempt status, where you can test your biomarker as part of the clinical trial and then would get approval. For many, many years very active in this particular part because it requires a very robust genomics assessment and validation as well as normalization in every step. And the real world context is that even when you do research, you're sort of sitting here. We now are doing these clinical trials and we're moving into the regulation. And I can give many, many other talks how this actually gets into the clinical arena and to convince CMS and payers and professional organizations that it's good to actually use what we're all here producing. So thank you very much.
You have several cases where you've taken multiple cores from these biopsies. So are any produced sufficiently good quality material that you could actually assay them all and look at the stability of the profile across all three?
A good question. So in principle, yes. Have we done it? No. So there's constant the challenge is to take these biopsies and do such a study, or to wait and use our biopsies for yet another extreme wells. Like now we are, like many people are working on cell-free circulating DNA tests. So that actually, which we are really in favor of. So we have plasma of all of these patients in many different, but we need then to do whole genome deep sequencing on every core. So we have interested parties who actually want to do that with us on every core. So then we save that core for that particular reason. So there's all the time a challenge. What do you want to do? And now with these cell-free circulating DNA, for instance, that requires 4 to 6 mil plasma, and we have 4 to 6 mil plasma. Because this, I told you, we have like 30,000 files. So we should have actually had a little bit more plasma. So my latest proposal is that just one of these arms will spend all of the plasma on this cell-free DNA testing, because that's what they require. But that's sort of always a trade-off. So yes, we could do it, but we haven't done it.
When it comes to ctDNA screening, it really depends on how deep you'd like to go.
Yeah.
There is some information to be gained, even if you use alpha field. It's a matter of philosophy, I guess.
Because what we actually, I alluded to that earlier, we actually very well set up. So originally drugs are tested and you need in cancer to wait 5 or 10 years, depending a little bit on the cancer type. But in breast cancer you used to need to wait 5 to 10 years because something got approved. With this pathological complete remission as an endpoint, it's a 6 months time frame. But actually we see on our MRIs and in our biopsies that after 3 cycles, 30% of patients have no tumor left. So they should continue, but we also can model that some other 40% is very likely to not benefit. So why give these patients another 4 months of the drug where we already know that they're not going to benefit? You would want to change. And so I think also circulating cell-free DNA will be the test to add there. So we are building our knowledge to get that time point solidified.
Yeah, we should probably talk because we have some algorithmic
Yeah.
infrastructure to do.
We don't have all the data yet, but it costs also a lot of money.
When you do the adaptive part of it, when you take the data and feed it back into the randomization probabilities, what are you optimizing for? Sort of the best outcome, or learning more about the experiment? In sort of machine learning, it's called an exploration exploitation trade-off.
So the first layer of that analysis is set to predict a successful subsequent trial with 300 patients, where you would give 150 the drug and 150 would get only standard of care. So that's the level where it's set. Now that we have added our qualifying biomarkers, we're actually setting it up to find the best combination that gives the highest probability of response. So that's a qualifying layer, but not the graduating layer of the drug. Does that answer your question?
So you're optimizing for the probability of getting a successful trial in terms of either qualifying or not.
Because what the FDA has said, you need to have 2 subsequent trials that show the same thing, and then they will give the drug accelerated approval.
So you talked about selecting drugs based on stratification of cancer subtypes. What prevents us from treating every patient as a single subtype and just finding a drug with the best therapeutic window on a cell culture derived from this patient?
People have done that actually, so there have been a number of companies that did that. So you could send a biopsy with living cells to that company, and they would test like 30 drugs or maybe 50 combinations. And in some cases it works, but in many it doesn't. So that has never been proven to be successful to predict response in the patient. In some cases yes, but in many no. So if you ask me, I think that what is most needed is an inpatient imaging marker, response marker, so that you early on can see if somebody responds to a drug. Like all of I'm involved in a large consortium that looks into metastatic cancer patients. So there are a lot of studies with all of the large cancer centers across the world. 7 are currently participating to combine all their data on, usually it's extensive targeted exome sequencing with cancer type and drug used. So that led in many cases to off-label drug use, like BRAF V600, which is a frequent mutation in melanoma and lung. If there's like one patient who had that mutation in breast cancer, who is metastatic, who has had all sorts of treatments, so are you going to give that breast cancer patient this melanoma drug, yes or no? And in some cases it will work, and others it will not. So you need to combine all of that data to understand what is the context when it works and when it does not. And so the American Association of Cancer Research is setting up a database that will be accessible by researchers to actually further research all that data and to recognize patterns. So currently it's only the variant calls, but I think if everybody starts to become comfortable in sharing the data, because that's a big deal, I think they will only also start to share all of their underlying next generation sequencing data. So then I think you can come to sort of understanding the algorithms, when is it efficient to use something for one patient as you propose. But there are a lot of toxicities with these drugs, so people are hesitant sometimes.
Follow
One
up
more
to the same question. What about xenografts?
The xenografts are also being used, which also works in some cases. But I think ultimately you test it in the patient, so that was also the vision of I-SPY 2, that we need, in a safe setting, start to test these drugs in patients.
Also, it takes a while to grow.
Yeah.
handling combinations? Because even if you have, say, 8 different drugs in the trial, there are obviously 56 possible combinations.
Yeah.
And there are, of course, safety as well as efficacy considerations. Do you test them individually, or?
Yeah, so the whole I-SPY 2, which is a phase 2 trial scheme, actually has a whole I-SPY phase 1 group of clinics where the investigational drugs are tested in combination with the standard of care taxanes. The FDA so far has said that if each of those individual components has been tested with taxane for toxicity, then it can be used as a combination. But that's an ongoing discussion probably.
Maybe we should take the remainder of the questions offline.
Yeah.
Thank you again.